Okay, class. So in part B, we're going to start to look at. Sorry, class didn't have the microphone set. So in part B today, we're going to sort of look at how the motor proteins and the cytoskeleton sort of come together to elicit some other cellular processes that we'll look at. So start off, we're going to just sort of take an overview with cilia and flagella. So unicellular organisms and sperm use flagella to move. And a fair number of epithelial cells actually have cilia on them, and so do other micro or unicellular organisms like parameciums and things like that. And so here's basically how the cilia function. Okay, you have a power stroke where it gets pushed and it's sort of force generating, and then it sort of curls back on itself and moves forward for a second power stroke. Okay, and that allows the sort of like paramecium to move and sort of swim around. And you can almost sort of think of it like a swim stroke if you want to sort of use that analogy. In the case with flagella, they work a little bit different. They just sort of swing back and forth. And here's basically an EM of a cross section of Chlamydomonas, which is sort of considered sort of the photosynthetic yeast. And here's its flagellum. It sort of can sort of move around and swim in solution. And it turns out that you have these microtubules here, and you have basically dynein in between them, and they actually cause the microtubules to slide back and forth, and that causes them to bend. Okay, and then that bending can actually create some movement. And here's sort of a schematic of how it looks. You have basically your microtubule bundles, the A and the B, and then you have your dynein mixed in here, and then there's some other proteins, and then you have your radial spoke proteins, and then it's basically encased in a plasma membrane. Okay, and so the ciliary dynein, can basically move. Here it is. It's sort of got that triple ATP domain in it that sort of functions like a ratchet, and it moves on the microtubules to basically cause them to bend. Okay, and it bends this axonine here, and you can sort of see it's going to move them in the plus direction. And when you have these linking proteins in them, it causes actually a bend. It can't just move. Okay, so this is basically how flagellum actually is moved back and forth. It sort of bends and sort of slaps back and forth. So I don't put a lot of emphasis on this, you guys, but it is good to know if you're interested in basically looking at it and understanding it more detail. But one thing that I do really want you guys to know is how the microtubules and the motor proteins sort of come together to form the mitotic spindle. All right, and when we cover the cell cycle, we'll look at this in even more detail. But for now, you just have to sort of be able to identify all of the different microtubules that are present in a mitotic spindle. You have your astral microtubules that are coming off the centrosome, and they radiate out in the plus direction. Then you have your kinetochore microtubules that are shown here in blue, and these actually attach to the kinetochore on the sister chromatids. And then you have your interpolar microtubules that are sort of cross-linked with kinesin motor proteins. And there's also motor proteins that are down near the centrosome. Okay, and so these motor proteins have various functions where they sort of basically push the centrosomes apart after cell division and after you've gone through basically the metaphase to anaphase transition, when the microtubules start to basically be pulled apart. So once you're in sort of anaphase and then you get into this next phase, which is called telophase, the centrosomes actually get pulled and pushed apart. Okay, and it's the motor proteins in between these interpolar microtubules that actually force that to happen. Okay, so here's just a great phase contrast image of a mitotic spindle in metaphase. So here you can see all of the chromosomes are actually aligned on the metaphase plate. Here you can see the centrosome, and then you have your kinetic core and interpolar microtubules sort of spattered in here, and then you have your astral microtubules coming off on the other side. Here's an immunofluorescent images of it. So once again, your centrosome, then you have your kinetic cores sort of shown in red here, and then the DNA, the sister chromatids aligned on the metaphase plate is actually shown here in blue. And like I said, we'll cover a lot of what's going on in all of this when we cover the cell cycle, but it's really important for you guys to know your astral microtubules versus your kinetic core microtubules versus the interpolar microtubules. Okay, now we're going to look at movement of a cell with lamellipodia. And we sort of started to talk about this last time, where you basically have actin polymerization in lamellipodia, and that actually causes what's called a protrusion. Okay, so here's just a cell, and it's crawling along a surface, and the first thing that happens is you get actin polymerization at the plus end, and this is done by formins and profilins adding actin subunits onto the plus end, and that leads to a protrusion. Okay, but you then actually have to sort of pull up the rear, okay, and have basically what is considered a contraction, and that's done by the myosin II motor binding to other actin filaments. Okay, so you'll have this protrusion where you have polymerization of the actin, then you'll get a new attachment which gives the cell some traction with these sort of focal contacts, and then the myosin II will sort of pull up the sort of the rear of the cell and cause it to contract, and then it can basically undergo the next round of protrusion. All right. And we can sort of see this in this sort of time-lapse image here. Here you have a keratocyte, and it's sort of protruding on this end, and then it's being sort of pulled up in the rear. Okay, and here's a nice EM of it. And you can see here, sort of at the leading edge, is where all of the actin polymerization is occurring. And in the sort of the back of the cell here is where all of the myosin II is contracting to basically pull the tail forward. Okay, so when cells move by basically forming lamellipodia, it's sort of a two-phase event where you have polymerization at sort of the leading edge, and then contraction and pulling together of myosin II at the trailing edge. Okay, and so here's basically just a really nice immunofluorescence of that. The actin is stained in red here, and then you have the ARP complex, which is involved in basically leading to the polymerization of the actin filaments really on the leading edge of the cell here, okay, as it's moving forward. And here's just a sort of a nice EM image of the actin filaments in lamellipodia. You can see them here, and if you sort of look at the numbers, you can, or look at the letters here, you can actually get close-up views of each of them. Okay, and so the ARP complex is leading to basically polymerization of the actin filaments here, and it's sort of pushing it and helping to push the cell forward in a protrusion event. Okay, and that actually is part mediated by and held together by cofilin. So here you have just a better image of that. This time it's stained red in actin and the cofilin in green. And you can see at the very, very leading edge where you only have the actin polymerization occurring, and then sort of stabilization behind with the actin and cofilin. And here they are. You have the ARP complex, and we sort of saw an image like this before. And then you can get the capping proteins and the cofilins here to help stabilize different things. And you get the polymerization on the end, and then there's a net sort of filament disassembly at the sort of behind that leading edge, and then the actin monomers can diffuse and basically polymerized on the head. Okay, and then the ARP complex likewise are are sort of recycled and moved to the end to basically help the nucleation event each time a new actin filament has to branch off a previous existing actin filament. Okay, so in general, this is a process where you have cofilin and capping proteins and the ARC complex and actin polymerization to lead to the protrusion event. And then in the contraction event, what you have is a bipolar myosin molecule that sort of pulls the actin together, and that actually helps pull the back of the cell, the trailing edge, and it pulls it together to help it move forward. Okay, so there's as much forward movement as there is rearward movement in a cell as it's crawling along a surface by using the lamellipodia to sort of reach out, and then the myosin 2 and actin to contract to sort of move the cell forward. And so here you can sort of see that you've got the leading edge of a cell here, and if you use this chemical compound cytoclasin, it actually caps the actin at the plus end, and what you end up getting is rearward movement. Okay, because you can no longer have the protrusion event, you just have the contraction. Okay. Here's the chemical structure of the cytoclasin. Oh, just before I go forward, make sure you guys understand the whole movement of lamellipodia in a cell, then using lamellipodia to sort of move across and move out. And we sort of saw this kind of happen in that first video that we saw in class, where you had the leukocyte transmigration, and it actually sort of moved on in by basically forming lamellipodia and crawling into where it needed to be during the site of infection. But once again, here's the structure of cytoclasin B. It will cap the plus end of an actin filament and just prevent further growth. So it's kind of useful chemical compound in the laboratory to study this process. Here's a beautiful EM of lamellipodia forming, and the cell's actually moving in this direction of the arrow here. You can see these great little leaflets that are sort of protruding out, and they'll reach down and attach. And this leads to cell traction, right? The attachment molecules, which we'll cover later when we're talking about the extracellular matrix, these can actually attach to a surface and pull. And so if you put down a silicon rubber sheet and you put some cells on it, they'll actually have a little traction and pull, and you can sort of see them pulling by these wrinkles that are occurring in the cell. And in the case with like cells in a multicellular organism like a mammal, our cells actually bind to other cells and also to the extracellular matrix. And one of the proteins in the extracellular matrix is collagen, and you can see these two fibroblasts. There's one on the top and one here on the bottom that's actually pulling this collagen fragment, and so it creates a little bit of tension there. Okay, now we're going to just switch a little bit to signaling and how cells change their largely their actin and microtubules by cell signaling. Okay, and that happens through G proteins, and the G proteins are basically the RAC, Rho, and CDC42 family members. Okay, so here's basically actin staining in a quiescent cell, and then if you take and you cause Rho activation by putting in a dominant negative activated Rho or a dominant RAC activated G protein or a CDC42 activated protein, you basically get different changes in the actin cytoskeleton. And these sort of Rho, RAC, and CDC42 molecules that are these small G proteins that are activated, they're pretty easy to make in the laboratory, and that sort of comes back to our understanding of the RAS mutation, which actually locks it in its GTP bound form, which is where it's active. Okay, so in Rho activation, RAC activation, and CDC42 activation here, you have the G protein locked in its GTP bound form, and it tends to change the actin filaments within the cell. And so here's how the signaling works. You guys don't really have to know all the details behind this, just try to get the big picture. Where you have rack activation, and then that leads to rack in its GTP bound form. So a GEF is going to basically do the exchange of GDP for GTP, and that'll basically activate a whole bunch of different pathways. And eventually what you'll get is branched actin web formation in the lamellipodia. Okay, and then that'll inhibit the myosin light chain kinase, and then you'll get decreased myosin activity to a sort of a lesser extent, but because you still need to pull the tail, but in the rearward movement, however, it's decreased myosin activity. Okay, in row activation, it's a little bit different. This will cause basically activation of the formins, and that leads to actin bundle growth, and then the row will activate the row kinase, which then actually can lead to increased myosin activity. Okay, and then you'll get integrin clustering and focal adhesion, and we'll cover the integrins when we're covering the extracellular matrix. In the case with CDC42 activation, we're going to go back to sort of our formation of the SHMU in Saccharomyces cerevisiae, where you'll get the mating factor bound to the mating factor receptor, which is a G protein coupled receptor. That'll signal through an activated G protein, the trimeric G proteins, cause exchange of GDP for GTP, and that'll eventually lead to downstream signaling where you have CDC42 activation, where it's bound in its GTP bound form, and then the rows will sort of signal to the formins, and you basically get polymerization of the actin, and you get the sort of polarized exocytosis and the sort of the SHMUing that we saw. Okay, and then just that also signals to WASP, WASP, and formation of the ARP complex, which also facilitates actin polymerization. And so once again, if you guys go back to the SHMU that we looked at the start of the lecture last time and the signaling lecture, you'll see that the mating factor is actually causing a rearrangement of the cytoskeleton, specifically the actin in Saccharomyces, which then causes the SHMU to form. Now, this same sort of process actually works for neutrophil migration. So it turns out that you can model this in the lab if you use this formal metlu phenylalanine, which is, you can add right near a neutrophil, and this formulated methionine, which is shown right down here, that's actually only produced in bacteria, and so when the neutrophil senses it, it's actually going to move towards that. So if you guys go back to that sort of images where you saw the neutrophil chasing the bacteria, it's actually sensing this formulated methionine here, and it's causing the cell to move towards it. Okay, so you'll have basically actin polymerization growing on the leading edge of the lamellipodia, and then contraction in the tail and the rearward movement. Okay, and so here's how it works from a signaling perspective. You have your chemoattractant basically being secreted by the bacterium. That's bound to a receptor, and then you can get RACT activation and actin polymerization, and then basically Rho activation, which can then lead to actin and myosin contraction in the tail of the cell. So you have protrusion and then contraction to allow the neutrophil to basically chase the bacteria around. Now, localized rearrangement of the cytoskeleton is also important when sort of setting up a synapse, be it a sort of neurological synapse or an immunological synapse. And so if you sort of look what's happening here, this is an immunological synapse where you have a T cell sort of associated with a target cell. There'll be a signaling reaction, and then you'll get localized actin polymerization between the two, and then ultimate formation of an immunological synapse. Okay, and you can actually see the centrosome actually moving down, or the centromere actually moving down here to help reorient itself. Okay, and by doing this, it can send the necessary molecules and transport the necessary molecules to the immunological synapse. Same thing happens sort of not only in fibroblasts where you can move things around. You'll have vesicles, and so here you can see the blue vesicles are bound with dynein, and the red little dots are vesicles bound with kinesin, and the green is the microtubules. And you can just sort of see by where the plus end and the minus are, the dynein is actually moving in the minus direction, and the red vesicles bound with kinesin is moving in the positive direction. Same thing happens with a neuron. You'll have your dendrite and your sort of synapse, and here's your axon, which can oftentimes be quite large, and you'll have the microtubules running down and vesicles moving between them. And this can be used to deliver neurotransmitter, as well as just growing the plasma membrane, and other things like that. And we'll sort of cover vesicle transport later on, but it's important to understand that the motor proteins, dynein and kinesin, are actually bound to these vesicles and moving them. Some in the plus direction, as with the kinesins, and then dyneins move them in the minus direction. And here you can sort of see neural growth cones sort of growing, and we'll look at that. And here's the axon right here. Then you have your little dendrites in your cell body that's present. And here's just a scanning EM of it to show the sort of the connections of the dendrites. Here's just an image of the vertebrate neuron. We've sort of seen this before, and it turns out that overall the cytoskeletal architecture of neurons is pretty complicated, right? And the dendrites and the synapses can actually change, over the lifetime of the nerve cell. And so here you can sort of see the dendrite architectural changes. And here you can see this sort of bleb that sort of protrudes out and it sort of stays constant, but the one sort of adjacent to it is sort of not there. Then it starts to grow back out again. And so you can see, just looking at this one image, it's pretty dynamic. And over the course of this 80 minutes, you can see major changes in the dendrite architecture just by looking at it. And so we'll look at this again when we're looking at nerve cells and long-term potentiation, but this is one of the ways in which connections are maintained to maintain memory and things, and memory and learning. So that's all I have for today. So the next class we're going to cover the cell cycle. So read chapter 17, pages 1054 to 1101 in the 5th edition, and 963 to 1010 in the 6th edition. And just as a reminder, the second exam is scheduled for Thursday, March 25th, and it'll cover lectures 9 through 15. So I look forward to seeing you guys all this afternoon, and I'll talk to you later. Bye-bye.